Imagine you have a child born with all 10 fingers and toes and is otherwise physically healthy. Then, around 18 months of age, the child starts losing eye contact with others, doesn't sit up on their own anymore, cries for no particular reason, and the deterioration continues from here. These are early symptoms characterizing a disorder known as Rhett Syndrome. Rhett Syndrome is a neurological disorder that mainly affects young girls, since it is fatal in males. It is lethal in males since gene mutations that cause Rhett syndrome are found on the X chromosome, one of 2 sex chromosomes. Since males only have 1 X chromosome, mutations in this location are so debilitating, leading to death within the first year of life. Hence, the aforementioned symptoms tend to be observed almost exclusively in females, as they maintain 1 normal X chromosome to offset the effects of the mutation on the other. Currently, there is no cure, and available treatment focuses on alleviating the symptoms of the disorder. In 90% of cases, Rett syndrome is caused by a mutation in a specific DNA regulatory protein that is highly expressed in the brain. Since there are so many different proteins that modulate DNA function, let's imagine that DNA is a dictionary to which words can be described. In the cell, in this case we're specifically talking about brain cells, we call these writers, proteins that add important molecules to the genes. Additionally, we have editors who can erase and change the way these words are described. In the cell, we call these erasers, as they remove the molecules that can be bound. And last, but most certainly not least, we have the audience, you and me, who interpret the meanings of these words. In the cell, we call these readers. Arguably, it is the readers that are the most important, since if there's an issue with the reader, the message is not conveyed properly. This is where my research comes in. My research focuses on characterizing the regulation of the gene for this reader protein. The theory behind this is that if we can discover the normal patterning of this gene, we can compare this to the abnormal regulation that occurs in Rett syndrome, ultimately offering a therapeutic to correct this abnormal signaling. What makes this so important is that it paves the way for clinicians to treat the underlying cause of the disease. Moreover, studying this gene has implications not only to Rett syndrome, but a range of neurodevelopmental disorders affecting both males and females, such as autism spectrum disorder. Thus, one day, we can touch the lives of parents and children across the world who make up the 115 million people affected by neurodevelopmental, behavioral, intellectual disability. Thank you.